Hello students, as we move on to the next section of our syllabus, we are going to see what we are going to deal with in this section. Now, I need you to recall your knowledge from the previous classes where we had studied the tissue arrangement in the plant. Try to recall the terms like parenchyma, sclerenchyma and collenchyma type of tissues, and we had talked about cambium and whatnot. If you cannot recall, I would advise you to go through those lessons again, because now we are going to see the arrangement of that particular tissue system within the plant, and that particular arrangement would comprise what we call as plant anatomy. Now, our section of multicellularity in plants deals with two sections. One is plant anatomy and the second one happens to be plant physiology. Now, why do we study plant anatomy? So that we can study the physiological functions in the plant in a better way. For that, we need to see how the cells are arranged inside a plant. So, we are going to see the topic that is plant anatomy. You have studied that what type of tissues are present in the plant. We are going to discuss that as well in case you don't remember. And as I told you, go through the lessons again so that you can get a better idea of whatever we are talking, because by the end of the lesson, what the aim of lesson is that we get to know that what is the tissue arrangement inside the plant, so that we can in a better way understand that how the physiological functions would take place, understanding that anatomy in a better way. Okay, now in plant anatomy, what we are going to deal with in the syllabus that we are going to discuss, first thing is that we had talked about tissues. Now we are going to talk about sections of root and stem. Okay, roots and stem. This is the first topic that we would be dealing with. Okay. Now, because the plant mainly comprises of roots, stems and leaves, so first thing that we are going to deal with is section of root and stem. We know that there are two broad categories of the plants. One is monocot and other one is dicot. This probably is revision of previous understanding only. So, one by one, we are going to see the section of monocot root and monocot stem. In your syllabus, you have to deal with transverse sections. We have enough lectures dedicated to understanding what a transverse section is, what a ventral section or vertical section is. Then in the case of dicot plants, we are going to see again the section of root, a dicot root and a dicot stem. So we are done with two parts of the plants. We are going to see how they look at microscopic level. So basically, in your syllabus for this class, the anatomical part would deal with microscopic understanding of the various plant tissues, their arrangement at the microscopic level in the monocot root, in the monocot stem, in the dicot root, in the dicot stem, and for understanding that arrangement, we'll have to make sections of the given roots and stems. You're going to see how we make that section, and probably you would practice that in your lab practice as well. The second thing is, because we have dealt with roots and stems, now we have leaves as well, which vary in both dicots and monocots. So we are going to deal with sections which is vertical section. Over here we were talking about transverse section. This TS stands for transverse section. We'll have vertical section of leaves. Okay. Now the leaves would be again dicot and monocot leaves. Both will be differentially cut and their sections would be observed under microscope. Then the third topic that we would be dealing with after this in the anatomical aspect, that is of secondary growth. Now when I talk about secondary growth, you need to have an elementary idea that what this secondary growth is. You have seen in dicot plants that there is increase in girth. We had talked about this in the previous class as well, that the dicot plants, they show an abnormal increase, not abnormal, but a different increase which is quite different from the monocot stem. The dicot stem shows secondary growth. Now, what is this secondary growth? Try to recall the drawings that you used to make in your lower classes of a tree. So this particular tree started from a slender plant which had a slender stem, and later on it became a huge trunk, and it transformed to this tree. Now this trunk has increased many-fold in girth. How this trunk has increased from this slender stem to a trunk, that particular process is known as secondary growth. And in our syllabus, we are going to observe this secondary growth taking place in both dicot stem and dicot root at the microscopic level. Alright, so now you can relate that why we studied those parenchyma, sclerenchyma, why we are going to study the arrangement of these cells or tissues to be very specific in a particular stem, their sections, because we have to ultimately understand that how it grows in girth. Girth is breadth basically. So the increase in girth of a plant, that is the stem increases in girth, that is because of secondary growth, and that secondary growth takes place at the microscopic level. For that we study the sections, and when we are done with it, we'll be over with the plant anatomy. So keep in mind that what we are going to study, we are going to study all these topics. First one is the sectioning of the root and stem, both monocot and dicot. Then there would be sectioning of leaves. We are going to understand what would be the arrangement of various tissues in the given leaves. Then we are going to see the secondary growth in dicot root and stem, how it takes place at the microscopic level, keeping in mind the tissue arrangement, because ultimately it is the tissue which is responsible for the secondary growth. And with this we'll come to the end of chapter. This would take around 6 to 7 lectures which you have to listen to very attentively. I hope you'll enjoy this topic.